Hi, everyone. Good morning or good afternoon. I'm a bit lost in the time zone right now. So indeed, I'm going to discuss today about EV's biodistribution, meaning how EV spread in multicellular organisms, starting from a point that can be either the secretion point for endogenous EVs or the injection point for exogenous EVs. I'm not going to make an exhaustive overview of the field, but rather I'd like to point out the few things that we know and the many things that we don't know, and you will see that this field or this subfield is really in its infancy. So when we see, okay, this is not moving. It's supposed to be a movie there, but it was working just in the rehearsal, but that's okay. So you can imagine here that you see EVs that are moving in the circulation of zebrafish. So when you see that, or if you think about that, or well, let's think about all the EVs that circulate in your own body fluids right now. When you think about that, then necessarily you can wonder where they're coming from, what do they contain, where are they going, and what is their function. And importantly, what are the mechanisms that make them arrest on a special place and perform their function. And it's really important to understand that, both in terms of fundamental and translational research, because nowadays there are more and more studies showing that EVs are involved in inter-organ communication, both in physiology and pathology. So you have an example here of adipose tissue EVs. But also, as we just heard, EVs are promising therapeutic tools, and therefore we need to understand their biodistribution, their pharmacokinetics, and eventually to modulate their destination and force them in certain places. So obviously, the challenge that we are facing in this field is that we are trying to track nanometer scale objects in tissues or organs that are centimeter range or even meter for the tallest of you. So it's extremely challenging, and for that a number of animal models were developed over the past years, from C. elegans to monkeys. Eventually, one day, kangaroos could be useful, especially for those who struggle with tail vein injection, as I do. So in the most simple model organisms, but also in the embryos of some of these organisms, the animals are translucent enough for us to use fluorescence microscopy, and in living organisms, I mean. And for this, we reach a resolution that is close to a single EV resolution or to a big EV. Whereas in mammals, for instance, the skin is perturbing the diffusion of light and prevents the use of such technology. And in general, what we can observe in these animals are bulk EV signals. So there are a large number of imaging technologies that have been used to try to look for EVs in animals. I don't have time to go through all of them, but we can eventually discuss the pros and cons during the roundtable. What you need to keep in mind is that the methods that have the highest spatial resolution are usually not applicable in living animals. Whereas the methods that can be used in vivo, and because they have a very high signal-to-noise ratio, usually have a very low spatial resolution, at least too low for detecting EVs. So this is why we observe a bulk EV signal. Associated with these different methods are different labeling strategies. Again, I'll be very fast, but these are either indirect labeling strategies where you will label the secreting cell, or direct labeling strategies where you will label the EVs. Charles discussed that before. So, of course, when you want to track endogenous EVs, you will need to go for cell engineering. Whereas if you want to track exogenous EV biodistributions, then you can use both type of methods. So I'll just finish this background part with two examples. One on your left is performed in zebrafish embryos, where you can see that we can detect extracellular vesicles with subcellular resolution, whereas in the mouse example in the right, you can see that we stay at the level of the organ to detect EV signal. And if we want to go deeper, then we need to go through ex vivo methods, as for instance here immunofluorescence on lung sections, or for instance cytometry. It's not the only difference between these two studies. The other one is that the one on zebrafish was performed using transgenic zebrafish embryo, which expressed a fusion protein between CD63 and a fluorescent protein, whereas the experiment in mice were performed by single injection of bolus of EVs in the circulation. So each method has their pros and cons. And depending on the question you ask, you have to properly choose the model that will address the question. So a few things to keep in mind in terms of EV biodistribution experiments. The first thing is that what we are following once we are looking at our animals is actually the probe, not the vesicle. And it's really important because in some cases the probe can dissociate and sometimes very quickly from the vesicle. And this has been shown, for instance, for lipophilic dyes that you apply on the surface of EVs. In vitro, at least, some of these dyes have relatively low stability on the EV membrane. But even for the stronger labeling, after a certain time, you can never be sure that you are still following the vesicle, right? The second point that seems really important, and it's true in particular for biodistribution studies, that when you label your EVs, it might impact their morphology, so their size, their surface, their charge, but also their behavior in vivo, so their biodistribution, we'll see an example of that, but also their uptake and their function, and this needs to be properly checked with the appropriate controls. So finally, well, I think you're all aware because it's 20th time we say that today, but it's important to report all experimental details. So in this case here, it will be labeling protocols, injection routes, but also imaging parameters. And all of this is very important for biodistribution because actually experimental parameters will affect where your EVs will go, and this has been shown by different groups. You have an example here from a paper by the group of Samirel and Andalusi, showing that comparing 3 different injection routes in mice, intraperitoneal, subcutaneous, and intravascular. And you can see that the same organs are targeted by the EVs, but in very different proportions. Similarly, if you use EVs from different cell source, you will also have different biodistribution. In a more recent study by the group of Charles Lai, they compared 2 different labeling methods, a breath-based method and a lipophilic fluorescent dye. And you can see that the results are very different with the same type of EV. So EVs will distribute differently. Similarly, if you inject different concentration of EVs, you will also obtain a different biodistribution. Okay, so that said, what do we know about the journey of EVs from their secretion point? So the first thing is that EVs needs to reach the circulation. So it's easy for cells that are facing the blood flow, such as endothelial cells, or in this example here, yolk cell from zebrafish embryo. So then the EVs are directly secreted in the circulation. But we know that in the circulation, we can also find EVs that have been secreted by cells that are not in direct contact with the circulation. And in this case, we have to imagine that EVs will have to cross the extracellular matrix and cross the endothelium. And the processes involved here are so far unknown. As an alternative road to the blood circulation, several groups have described EVs traveling through lymphatic vessels, and this is the case here in these examples for melanoma EVs, both endogenous and exogenous EVs that can be found in lymphatic vessels and accumulating in lymph nodes. Once in the flow, we know that extracellular vesicles are circulating with the blood flow, but they also have a very short half-life that can, sorry, so there should be some movies as well. Okay, yeah. Yeah, so the half-life of EVs have been reported to be as short as a few minutes up to a few tens of minutes. So EVs in the blood flow are very quickly taken up, mostly by endothelial cells, but also by peripheral blood mononuclear cells, so immune cells in the blood, such as, for instance, patrolling monocytes, but also lots of different other types of immune cells. And as we've heard just before, the half-life of EVs in the circulation can be controlled and manipulated, for instance, by playing on the receptor CD47, the so-called don't eat me signal that will extend the half-life of EVs in the circulation, or by covering the EVs with polymers, as Marie just mentioned. Okay, so now how do EVs stop and how do they accumulate in specific organs? We know a few things about that. We know that adhesion receptors present at the surface of extracellular vesicles seem to be important. The first ones to be properly described were the integrins by the group of David Leiden almost 10 years ago in a cancer context, and then other adhesion receptors were shown to be involved in EV's biodistribution. And here is another example from Charles Lai lab where the knockdown of CD44 will alter the biodistribution of EVs in mice. So this is all very nice, but there's one thing that is completely missing from the picture here is the ligands of these receptors, which would make sense. Eventually, you can imagine that different organs or different cell types will express different proportion of these ligands, but so far we don't know them. Other natural modification of EV surface can be involved in the biodistribution. This is the case, for instance, of the level of glycosylation, but also the presence of the corona that forms naturally around extracellular vesicles. So this is it for targeted biodistribution, but it's also clear that there can be passive accumulation of EVs in some organs, for instance, in clearing organs in physiology, such as the liver, but also in pathology, EVs can accumulate in tumors due to the PR effect. Eventually, they can be taken up by non-specific receptors, such as scavenger receptors. In some cases, circulating EVs were shown to cross very tight barriers, such as the blood-brain barrier. And in this particular paper, they showed that EVs can cross the endothelium by transcytosis, meaning being taken up at the apical side of the endothelium, trafficked through the endothelium, and released on the basal part. And then finally, once in the tissue, EVs will eventually target receiving cells and transmit their EV signal. How that works in vivo is so far very poorly understood. So just a word now on xeno-EVs. So the same type of biodistribution studies can be applied to EVs from different species, for instance, bacteria. On the left, you have a very nice example coming from a paper from the group of Stephen Moma, where they grafted in the gut of mice bacteria that release OMVs, so EVs, that contain the CRE recombinase, and they use reporter mice to track the cells that have received CRE recombinase, and they found such cells in many different organs of the mice, suggesting that maybe bacterial EVs from commensal bacteria in the gut could reach these organs. On the right, you have a more simple example where pathogenic bacteria EV are injected in an animal, here a rooster, and followed in vivo. So, as you've understood, the gaps in knowledge are many. The first one, probably an extremely important one, is that we still don't know what is the natural secretion rate of EVs by different cell types or different organs in different contexts. And it's very important, I think, to understand that both in terms of fundamental perspective, but also to properly calibrate the experiments where we inject exogenous EVs in animals. So then what are the EV roads? We know a few of them. What are the parameters that affect these roads and the biodistribution of EVs? We know a few of them. We still don't fully understand how EV reach specific organs, but maybe here there is a parallel that can be drawn with two very ancient theories in the cancer field, postulated in the late 19th century and early 20th century by Stephen Paget and James Ewing. The first one is so-called seed and soil theory, which says that metastasis will form in the organs where the environment is favorable. And the second one is more of a mechanistic theory, saying that metastasis will form in organs following specific vascular patterns. And probably there is a parallel that can be made here with EV's biodistribution. We can eventually discuss that later. Finally, what are the consequences of these different patterns of biodistribution on the function of EVs? And I mean more precisely on the balance between EV degradation and EV cargo delivery. This is fully unknown yet. So what could help us fill these gaps? Probably we will need to generate new animal models that will be specifically adapted to the tracking of EVs in vivo, such as, for instance, the Exobo mouse model developed by the lab of Sonia Melo recently, that allows for the tracking of pancreatic EVs in mice. In addition, we will probably need to develop new probes, for instance, multimodal probes that would allow to track EVs at different scales, from the subcellular up to the organism scale, or smart probes that would allow us to determine whether EVs are internalized or not. Okay, and with that, I will finish here, and thank you for your attention.